Hi everybody, my name's Kip Ludwig, and today's module is about preclinical safety and efficacy studies for neural interfaces. To tell you a little bit about myself, I was the senior research scientist at a company called CVRX. I developed a minimally invasive implantable neural electrode to stimulate the baroreceptors to treat hypertension and heart failure. I did the supporting preclinical work, both benchtop and large animal safety and biocompatibility studies, to take this to an IDE, to human trials in Europe, to CE mark, and eventually to FDA PMA approval. After that, I went to the National Institutes of Health where I was a program director, and I actually led the translational devices programs that related to neurotechnology. Since then, I've been a consultant for many of the companies on their own preclinical testing plans for neural interfaces for safety and efficacy. I am also an associate professor at the University of Wisconsin-Madison. I am the co-director of the Wisconsin Institute for Translational Neural Engineering, and my own lab within that structure actually specializes on both benchtop testing for FDA approval and then large animal preclinical testing in support of FDA approval as well. So, talking about all the scope of benchtop testing and animal testing for FDA approval is really beyond what can be done in 40 minutes. So I really have three goals and three topics for this lecture for you. The first is to discuss about what are the general goals of benchtop testing in animal studies to support FDA investigational device exemptions. The second is to direct you towards important resources and guidances from the FDA and elsewhere to serve as templates for developing your overall testing strategy. And frankly, that's going to be quite specific to the device and the therapy that you move forward. So the goal is to kind of give you the tools to be able to understand what is necessary. And then I'm going to talk about some of my own experiences and really the common mistakes I've seen not only made by myself and others I've worked with, but through my NIH programs where we saw dozens of FDA IDE and FDA pre-submission meetings and saw both some mistakes that can be avoided as well as best practices that will really help you avoid those things. So the first topic is what are the goals of benchtop and animal studies to support FDA investigational device exemptions, otherwise known as an IDE. And there's really two myths about preclinical or non-clinical testing to support an IDE and about FDA market approvals. The first myth is that benchtop and animal studies prove a device is safe for human use. And I'm going to talk about that in the next couple of slides. The second myth is that if a device is approved by the FDA, that it is safe, that it has no risk associated with. Neither of these statements is actually true. So the truth of the matter is, even with a lot of great non-clinical and preclinical testing, and even with a lot of experience, devices that are implanted in the body do fail sometimes. They do have risks associated with them. What you see on the screen in front of you is data from the follow-pace study. Now, really the predicate for a lot of neural interfaces, certainly the predecessor, if not predicate, is the cardiac pacemaker. There are about 900,000 cardiac pacemakers implanted worldwide per year. The first implants were in 1958, and there have been follow-up studies. One of the most famous is the follow-pace study, which was published in 2007, which actually followed a large cohort of people implanted with the cardiac pacemaker. And what is important to realize that is even though that these pacemakers are well understood and have been implanted for years and years, there is still a 12.4% overall complication rate at 2 months post-surgery. These include issues with the leads. These includes issues with the surgical pockets, including infection. And about 4.2% of these actually require reoperation. At the 1 year point, there is an additional 9.2% rate of long-term complications as well. So that's a very significant amount of complications and significant safety risks that do occur with a well-established technology. Now, the rates actually for the vagus nerve stimulator, the spinal cord stimulator, and deep brain stimulators are higher. These are not only less established technologies, not implanted nearly as many, but also due to their the local anatomy that they're being implanted, to some extent, there may be more risk of trauma and more significant trauma. Another important facet above the reported surgical adverse event rate and other complications that occur that are surgical or due to recovery from the surgery or due to stimulation, these are complex systems as well. So the vagus nerve stimulator from LivaNova, even though it's been FDA approved since 1997, has had 5 recalls in recent years. This can happen for many reasons. You have parts from dozens, if not hundreds, of suppliers, and sometimes they change their manufacturing process. So there are very subtle ways where you can run into a problem that actually creates a critical error. In this case, the one for the Sentiva system was viewed as a Class 1 recall, which actually means there is a significant risk of injury or death to the patient that they are trying to avoid. So what I find very useful is trying to understand the FDA reviewer mindset. Your goal really is to give them information to address concerns that they might have. And so it's really important to understand your audience. And as I went from having done animal experiments in my own lab to doing animal experiments in support of FDA approvals to rolling out devices to be used at 40 clinical centers as part of a pivotal trial, I learned things in terms of different issues that crop up that are difficult to anticipate without this experience. And so I'm going to give you a couple of examples, but I'd like to stress that the FDA sees thousands of devices a year. And so they have a lot of experience seeing little issues that crop up as you move to the next step that you may not have anticipated. And so it's really good to understand their mindset as you get into complicated issues like shipping that might create a problem, or how differences in your supply chain with one module might actually impact the entire system and other modules and how they function. So one illustrative example I like to use is a simple shipping issue. This is actually an electrode that I designed that's coated with titanium nitride. And I actually went through several coatings to attempt to improve the stimulation safety limits for these electrodes. And what you'll note in the picture in front of you is that the coatings are imperfect. There's actually a couple of flaws in what is that black area is the coating, and under it is a platinum iridium electrode. Now these flaws weren't there when it was initially coated at the provider, but what happened is due to shipping and vibration during shipping and due to thermal cycling, some of the coating material that's supposed to help with the stimulation safety had actually fallen off. And so it's really important to think through the entire process for your device, how things are being not only manufactured, but all the processes that they will go through before they arrive at the end system manufacturer, which typically is you, and you are responsible for all problems that could have happened to any module or how they might affect other components of your system. So here's another example, and this is actually a different coating that was used that was actually much more robust with respect to delamination. And there's certain tests you'll actually do for stimulation safety to test for delamination, but this is an iridium oxide coating. And on the top row, it's quite beautiful, but one of the reasons why they don't only look at individual components, but they want to look at the full end system the way it's intended to be used in humans, is what you see in the bottom row. So we got electrodes in that were coated with iridium oxide, but to some extent we have to create a connection to them to go back to the implantable pulse generator. This was originally done with a weld joint, and specifically a heat weld. And what you'll see in the bottom row is the changes in those coatings from the provider after we did a heat weld. And what you'll, in this particular case, you see a marked change in color and ultimately a marked change in function due to being welded at a high temperature. And so we actually had to come up with different processes to create the equivalent of a weld to connect a wire to these electrodes for electrical connection that did not involve high heat. And so these two examples I think are kind of illustrative of the sorts of things the FDA has seen at a wide scale, and why it's really important to think through every step in your process, and how one component, when combined with another, could also create a problem or a potential safety issue. And so one of the guidances that I've always found to be incredibly helpful, this is available online and the link is on this slide, is an overview given by the Deputy Director Owen Ferris, and this is actually a slightly older PowerPoint presentation that really talks about the FDA and the IDE process and really talks about it from their point of view. And so key points from this FDA feasibility IDA review document is, first of all, the IDAs are primarily focused on safety. Although efficacy can come in to some extent, and we'll talk about that later, it is primarily focused on safety. Now, I'm not going to read all of the key points that I've put on this PowerPoint for you, but I'm going to talk about things that are really important for conceptualizing what's important in preclinical testing. And what you're really trying to do is convince the FDA, one, why is clinical really the next necessary step in the development of this? And two, they're looking at, is there appropriate mitigation of potential risks? Again, your goal isn't to prove that this device is perfectly safe and has no way of failing. Ultimately, you're trying to convince the FDA that the next logical step is going to humans, and the rewards of going now, without any additional information, outweighs the risks as they have been defined right now. So, there are several basic submission elements for an IDE approval. The first is just talking about the background of the medical issue that you're trying to address. What are the goals of the clinical study that this IDE will enable? And why this study will further science. To some extent, in an IDE, there may be still some scientific questions that you're addressing in addition to trying to develop a device towards market approval. You need to give a detailed description of the device under study, and then you need to talk about the previous studies, both preclinical and potentially clinical. And it depends, IDEs can support different stages of clinical studies as well. So you give a summary of the available data. You talk about why is a clinical study needed and appropriate at this stage, and then you outline what evidence supports the safety of the study device and the potential for the study data to be meaningful. Second, you outline, are there outstanding safety questions that should be addressed with preclinical data? It is important to note, the FDA does not require proof of efficacy in animal models for granting an investigational device exemption. This is a common misconception. Now, sometimes they will require it, but if there is a good argument to be made that it is appropriate to go to humans without proof of efficacy in an animal model, and I'll talk a little bit about this later, then it may be appropriate to do an IDE to enable an early stage clinical study, even though there is no proof of efficacy right now in an animal model. And this is really because sometimes animal models may not be appropriate at all to evaluate the efficacy of a device. And if the device itself has been shown to have a lower safety risk, it may make sense to attempt it directly in humans. It's also important to consider that devices, for many reasons, are different than drug trials. I had seen a lot of academic investigators attempting to do translation for the first time, get an FDA regulatory specialist to talk to them and help them with their FDA submissions, and they had come from drug world. And sometimes this worked out well, sometimes it did not. And one of the reasons is devices aren't drugs. And so in this guidance from the FDA, they do talk about how they are different from drugs that might be significant in terms of how you develop non-clinical and preclinical testing and how you execute a clinical trial. The first is that, especially for implantable devices, the trials tend to be smaller than drug trials. Second, although there are many different types of devices for many different reasons, some are very novel and have very unique function, and there's not a lot known about them, but there's also devices that go to clinical trials that are essentially Me Too devices. They may be a different riff on something that's been around for many, many years, that's very well understood, both in terms of its efficacy and safety. Especially for neuromodulation devices, which are my specialty, it may be actually difficult to create a blinded, randomized, and appropriately controlled study like you can with an orally administered placebo. In many cases, especially in the early stages, there may be device modifications that occur during the trial. So it might change, especially as part of the early studies and as part of you learn, and that to some extent, the device's function may depend on physician technique, which will get refined over the course of using in humans in the early stage studies. The endpoints can be highly diverse. devices can treat a vast variety of different diseases and disorders, but also can be used for diagnostic purposes. We'll talk a little bit about this later, but typically devices follow a three-stage path before they get a PMA approval, and that is for a novel device that has significant safety risk. So typically you start with an early feasibility study, then move to a feasibility study, then to a pivotal trial. You'll often see people discuss this as a phase 1, phase 2, or phase 3 study because they're familiar with that parlance from drug world, but it's actually slightly different, and there's slightly different goals for a early feasibility, and pivotal study in devices. The most important thing, though, is that the data that you're trying to get out of this, it's designed to support a reasonable assurance of safety and effectiveness for the marketing application. Again, an FDA approval does not mean a device is safe. In fact, in many cases, there's many known safety risks. The goal simply is to show that the known risks are fairly well defined and that the benefits of the device outweigh the known risks. So the language you see in front of you is drawn directly from the FDA guidance on investigational device exemptions. And there's a couple of key points I want to go over, but I don't want to read this to you word for word. I've highlighted most of the key points in red. As I said previously, the device evaluation strategy should be based on a risk-benefit assessment. It should include information needed to address significant safety concerns and support basic device functionality. So the way you do this is often in a table format, and you start out in one column with just the device attributes. Then in column 2, you talk about the potential failure modes. This is actually often overlooked by people who haven't done this much, and it's really the most important point in terms of how to prepare for an IDE. All of your preclinical testing, all of your non-clinical testing stems from a failure modes analysis. And in simple words, for each device attribute, you outline the type of problems or failures that might occur and could result in consequences to the device or study subject if the function or feature is not attained. So you go through each device attribute and you say what happens if it doesn't do what it's supposed to do and what are the consequences about it. Then you talk about what are the potential device and clinical effects of each failure. Then you talk about the device design information and how your non-clinical and preclinical testing stems for this is for each attribute and our potential failure mode, the information from internal or external sources to supplement the assertions that the functions or features will be attained and or the failure modes will not likely occur or will not be catastrophic if it doesn't occur. This is often referred to, there's two different ways of looking at that. One is a failure modes and effects analysis, where you outline each individual component and you talk about how if each thing's failed, how that can create a problem. And then there's a hazard analysis, which is more top-down, which on a 20,000 foot level, you talk about all the potential risks that might occur and their severity, and then you work backwards from that. Now then, your non-clinical device testing is really to mitigate these risks. You outline the bench laboratory, analytical, and or animal testing of the study device to complete the evaluation of each potential failure modes. And ideally, you're trying to get all probabilities of a risk occurring to be low and the consequences of that risk occurring to be low. That's not always possible, and many IDEs get approved even though there's maybe something with medium risk and medium severity if it does occur, but the benefits of taking that risk outweigh the potential consequences. The other thing to realize, although benchtop and animal testing is useful for helping define safety risks, it's not perfectly predictive. Many risks in low-end animal studies or risks that aren't identified show up as you scale to hundreds and thousands of patients, many different institutions, and working with multiple suppliers. So you don't have to catch every single problem. You want to do reasonable due diligence. There's a number of reasons why the animals might not be predictive of all the risks. Animals typically have a low sensitivity for side effects. They can't tell you if they feel uncomfortable. In many cases, the animals won't represent the range of human anatomy, and therefore your effect and side effect profile might change. Animals often don't represent the previous treatments that may interact with your therapy. This is especially important in neuromodulation devices, especially implantable, that are treatments of last resort. In many cases, the subjects in those studies have been using drugs that manipulate those same neural pathways for decades. And this is why the FDA, in terms of clinical studies, tends to stage the risk. After you've done the preclinical testing, the first study is typically early feasibility if it's a new device. And early feasibility will be under 10 patients for a certain amount of time because they don't want to put 100 patients suddenly at risk for something that has a lot of unknowns in the jumps from animals to humans. Then there will be a feasibility study, which is typically under 100 patients for devices, and then the pivotal trials. For neuromodulation devices, you'll often see 200 to 400 patients. So there are really 4 key components to how you mitigate the known potential risks that are intended to work in synergy. Some will be discussed in other modules in more detail. The first is quality management systems. A lot of what I've discussed for supply chains is really dealt with primarily through your quality systems management, and that'll be covered in another module. Second is biocompatibility testing. This is taking a passive implant and looking for things like leachables, bacterial endotoxins, and pyrogen testings. This is typically done in small animals so you can get to high end and do some really nice statistics as part of the evaluation. Then you have both benchtop testing and large animal testing, and they have different goals. The benchtop testing is really your longitudinal testing for longevity of key failure points for life of implant. Now, for an implantable, you're typically shooting for 10 years, sometimes 5, in terms of device longevity. The other thing you do in the benchtop that you don't do in the large animal studies because you might not be able to is detailed characterization of likely mechanism of failure, leveraging several tools that aren't feasible in animals. Then the large animal testing is typically your definitive sanity check. It's typically a 3 to 6 month study to assess the full final locked system, ideally manufactured in the same way or as close as possible as will be done in the clinical trial. And this is intended to assess how different components might interact as well. So you want to change as little as possible between your GLP definitive chronic animal study and the actual IDE. You're looking for both side effects and intended effects sometimes, but really this is a sanity check for the known unknowns and unknown unknowns that you can't identify simply through benchtop testing. This is looking at things like lead migration as a function of implant, lead wear and abrasion, tissue response, QMS issues, and several other things that anybody who's done a lot of large animal studies will know they tend to uncover. But you're still not recapitulating 10 years of wear and tear. Realistically, because of the costs of all of these things, the large animal testing tends to be 3 to 6 months and tends to be somewhere in the range of 6 to 8 animals. And the benchtop testing really does a lot of your projection of how well will this survive the lifetime of 10 years and yet still be reasonable for you to do in terms of cost and time. This is the end of topic 1. And there's really 2 questions you should be able to answer at the end of this topic. One is, what are the key criteria the FDA uses to review IDE applications? And second is, how is a failure modes and effects analysis used to inform the non-clinical / preclinical testing strategies? So in the second topic, because it's going to be impossible to talk about all the details of preclinical testing and what's in an IDE, is really to talk about important resources and guidances you should be aware of and general 20,000-foot overall concepts that you really need to know. Pretty much the most important general strategy is plagiarism is a very good thing in this world. What you really want to do is find the most similar devices and their testing strategies that they use to mitigate risk for a previous FDA IDE and copy that strategy as much as possible. So I've outlined two really good templates for your failure modes analysis and for the testing that's typically used to address that failure modes analysis. So one of the most important one is there is a guidance, and the link is on the slide, for the research and marketing applications for permanent pacemaker leads and for pacemaker lead adapters. The second guidance that I've included is the retinal prosthetics guidance. And this is actually really useful in terms of it's a more complicated system in terms of how the retinal prosthetic works. There are wireless components that are talking to each other to inform neural stimulation parameters based off of camera readings and things like that. So these are, to some extent, similar devices to the cardiac pacemaker, but they have some technology differences that are important to be aware of. It's also very good to kind of look at the differences in testing that occur when you're implanting something in the eye versus something in the heart. And so I'm going to talk about the benchtop testing and the animal testing, as the quality systems and the biocompatibility are in other modules. So if you look at these guidances, there's really several key tests that are part of the benchtop testing plan. So the major goal of the benchtop testing plan, as stated earlier, is one, longitudinal testing for longevity of key failure points for the life of the implant. You're only going to implant in the animals for 3 to 6 months, and so you're really trying to extrapolate where these devices will fail, or if they'll fail, when they're implanted for 10 years. And then you do detailed characterization of likely mechanisms of failure, leveraging tools not feasible in animals. This can be very important in terms of accelerated lifetime testing and also just extrapolating curves to show that is the likelihood of problems becoming worse over time, or is it actually plateauing? So there's several categories of tests that are often done as part of benchtop testing that will outline the design lifetime and performance durability tests. So design lifetime and performance durability tests really often deal with many of the mechanical failure modes. These can include hermeticity tests for your IPG and other things. It can be evaluation of coating durability, welding and bonding patency tests, flexion tests, connector tests. Then there are electronics tests. And one of the things that's often overlooked in an academic design is that for the most part, you're attempting to develop a device to have what's called single fault tolerance. This means you have to be able to show that if any one component in your device fails, that it's not going to create a significant safety risk or a significant efficacy risk, or both. And so many devices that weren't made for regulatory approval are not designed with single fault tolerance in mind, and often have to be substantially redesigned to actually pass single fault tolerance. And this can actually be a problem in and of itself, because that changes the cost of your device. So given insurance reimbursement rates, if you redesign the device and developing it for single point failure modes, might change the cost of it, you can actually change your business case and not have a viable business case. Then finally, another component of this is electrode stimulation tests. That's a specialty of mine, but I'm primarily just going to, in the upcoming slides, point you to really good resources on electrode stimulation safety testing. So here are 4 really good resources to both give you a general framework for both benchtop and animal testing, as well as some specifics on electrical stimulation testing, as well as the basic theories behind the strategies to characterize. So the top one, done by Rob Shepard, is a good general one that goes through both benchtop and animal studies based off of a number of devices. The next one is actually myself and a group of my colleagues outline what we did for FDA submission of the baroreceptor stimulator for benchtop testing and animal testing. The third one is a fantastic guidance and tutorial about general electrode characterization techniques, what are the theories behind it and how they work. And the fourth one is really the history of how the existing standards for the FDA for assessing tissue damage thresholds for therapeutic electrical stimulation evolved over time. So in discussing what sort of animal testing you will do, there's a couple of things that are generally true but not always true. And then even whether you need a large animal, isn't always true, depending on the type of device and what history with that device is already known. So what's most common is you often do your biocompatibility studies, which is just putting the implant in or a portion of it passively to look at the tissue reaction and other things, is often done in a small animal model, such as rodent or rabbit, and this is because you can do high-end studies with them and they're cost-efficient. The large animal model testing will really have two elements, and these can be done together or separately. And it's really looking at the safety and the efficacy. And I'm going to talk about those separately, even though these are sometimes done together. So the major goals of the safety testing. The one is a chronic study to assess the full final locked-in system, ideally manufactured in the same way as will be used for the clinical trial. And as I discussed before, this is really to address the known unknowns and the unknown unknowns. You're going to be implanting somewhere around 6 to 8 animals for 3 to 6 months in a large animal model that's typically relevant of the representative of the relevant human anatomy as it pertains to safety. So one of the important things that they're looking for, especially with new devices where this isn't well defined, is assess wear and tear issues at correct anatomical scale. For implantable electrodes, whether it's on an artery or a nerve, as you move around, especially a large animal, you put a lot of torque on them. You see a lot of failure at transition points between a pulse generator and silicone leads, or when the silicone leads actually change in terms of their size and lumen diameter, and that you have filler and things like that. Those are things that tend to fail chronically. This allows the FDA to see this at scale. You can assess the tissue responses for both active and passive implants, and that's important to understand to some extent your electrical safety issues. Even though you can do a lot on the bench, there are things that are difficult to control for, such as DC offsets and other things that might occur that can affect your safety profiles. You're often looking for side effects as a function of stimulation in addition to your intended effect. Now, there are some exceptions to a large animal chronic requirement under GLP, and we'll talk a little bit about GLP later. If the safety risks are previously well established due to similarity to an existing device, especially at the anatomy of interest, or there is no relevant large animal model. For certain neuromodulation targets and neural interface targets, there really is no relevant large animal to a human, then it might not be appropriate to evaluate some of these safety things in a large animal, but you have to make a case for that. So in terms of efficacy testing, this is where things are somewhat different than the safety testing. So first of all, the major goals of efficacy testing is really to convince the FDA there is a reasonable chance of benefit that offsets the known and unknown risks of moving to a human trial at this time. So the animal model, in terms of large animal versus small, for efficacy, it can be heavily dependent upon the therapy. Now, it's often efficient to use the same large animal for both the safety and efficacy testing, but sometimes it doesn't make sense to do that. For instance, in some cases, efficacy is done in a small animal model or based on prior data suggesting efficacy of something similar in humans. One good example of this is a lot of applications for pain. There may be an established rodent model for efficacy for pain, but not a large animal pain model that has been established. Therefore, it would be difficult to use a large animal to show efficacy in pain. So one of my examples for the baroreceptor stimulator, and this was, we were stimulating the carotid sinus bulb to activate baroreceptors to treat hypertension and heart failure. That's technically complex, you don't need to know that, but we actually did the efficacy studies in canines and the safety studies in sheep. And we did this primarily because it made sense and was justifiable. So instead of a bifurcation of the common carotid into the internal external that you see in humans, sheep often have a trifurcation and actually often don't have a pronounced sinus bulb, and therefore it kind of limits their relevance for efficacy. However, the size of their arteries and relative muscles, etc., is much more appropriate for humans, so they're an appropriate safety model. Canines conversely, have much smaller internal and external carotids, which make them, and their necks are much more flexible than ours. So it actually makes them an inappropriate model for safety, but they're actually a very good and well-established model for efficacy for carotid sinus nerve and baroreceptor stimulation and vagus nerve stimulation for hypertension and heart failure. So we wound up doing two different studies, one for safety and one for efficacy. And this is not entirely uncommon. So good laboratory practices, often referred to as GLP, will be discussed in another module, but it is important to know what portions of your testing plan should be GLP and what portions do not need to be. So overall, what is GLP? GLP is a quality system concerned with the organizational process and the conditions under which non-clinical health and environmental safety studies are planned, performed, monitored, recorded, archived, and reported. That's directly from the regulations. I'm not going to read the rest of this to you verbatim, but I want to point out a couple of things that I think were a surprise to me when I first did my GLP studies, but are really important to understand what they're intended to accomplish. So one is in bold and one is in red. The bolded one. So facilities conducting studies in accordance with GLP regulations are required to have independent quality assurance, QA unit, to monitor each study to assure conformance with Regulation 21 CFR 58.35. The QA unit is subject to routine audits by the FDA. I've heard several academics say their lab does GLP, and in many cases, if they're the ones who would be the sponsor for an IDE, it would be difficult for them to actually be the GLP facility for their own study. And most importantly, there has to be an independent quality assurance unit that's subject to routine audits to the FDA. So often things are done that are considered GLP-like. They are actually doing everything in accordance with GLP except for this provision of independence and routine audits. But the reason this is important is, simply put, the FDA cannot trust you. You are biased. You have a vested interest in this going forward. So critical data to inform safety must be independently generated and traceable to raw data locked and safe from potential manipulation. And the FDA will come in when you have an IDE submission or when you have a PMA or 510K submission and sometimes ask to recreate from this independently locked raw data key analysis that you used. So what does this mean, and what are kind of key aspects of GLP? So in GLP, you outline a protocol, but it's important to note that there are sometimes non-compliances and deviations from that protocol, even in a GLP study that's successfully used, but they must be carefully annotated and justified as non-critical to the safety outcomes by the study director, who's typically independent. As I mentioned before, raw data must have traceability and clear assurance it could not be manipulated by the sponsor. Now, this can actually require substantial work with the CRO for anything not routine to them. And so in my particular example, when we were doing things for hypertension and heart failure, where we were doing implantable wireless molar catheter systems to measure blood pressure and heart rate, we actually had to come up with the manufacturer and the CRO a way to store the raw data daily, annotate it, and lock it for traceability so that they knew we as the sponsors could not manipulate it. Because this is actually much more complicated than a standard study, GLP is typically a 40 to 50% markup at a CRO versus a non-GLP study due to this substantial documentation burden, daily annotation of the data in many cases. So one of the questions I get most is what studies need to be under GLP and what studies can be done non-GLP. So technically, GLP is only required for key in vitro and in vivo studies in which definitive safety information is being assessed. Sometimes supplementary safety information does not need to be under GLP as well. GLP is not required for discovery. It is not required for basic research. It is not required for screening. It's not required for any vitro studies where definitive safety characteristics of the products are not being assessed. Now, all of these should still be done in accordance with good scientific practices for rigor if included in an IDE application, but they don't require it to be done by an outside independent quality assurance unit that gets audited by the FDA. Now, the animal studies are typically GLP. And they're really intended to identify issues that benchtop studies may miss. We've talked about wear and tear substantially. Contaminants entering the supply chain as well does happen. You should be able to implant 6 to 8 animals for 3 to 6 months without a contaminant entering your supply chain. And I've actually seen this happen quite often in non-GLP and even GLP studies, predecessors, where things like silicone oils on the inside lumen of a lead leach out as fluid gets past the electrode, and then creates a contaminant that causes an immune response near the electrode. GLP is typically done at a contract research organization. If you're going to work through a CRO, you really should always do a non-GLP run-through with the CRO first. It doesn't have to be for the full 6 months, but you're really going to need to work with them so that they can use your product and get the data that's outlined for your IDE and do it independently, and so that they can annotate that data and lock the raw data daily. So, an important aspect of your testing plan is whether or not to have a pre-submission meeting with the FDA or multiple pre-submission meetings, often known as Q-subs, with the FDA to discuss your testing plan. Now, there are several different types of meetings, and these are going to be talked a little bit about in other modules, but it's important for you to know that, one, you can just have an informational meeting with the FDA. If you have a very novel device, you don't have to talk about your preclinical testing plan or even your clinical testing strategies, but you can just introduce them to the device. These meetings are quite short, and so sometimes it's useful to just get them thinking, get them familiar with it, and get them to maybe give you some questions about its functions, or so you can assess how they're thinking about something if it's very novel and something they haven't seen before. Now, it's common as part of these pre-subs to talk about your benchtop testing plan and your animal testing plan and appropriateness of animal models. You go in at risk if you submit an IDE and you haven't discussed previously your benchtop and animal testing plan. You complete all of it and you just submit it without talking to the FDA first. Now, this is not uncommon to do if it's something that's very well established, but if you haven't done this before or your device is novel, it's really good to talk to the FDA and get some feedback on whether your plan will be appropriate before you do it. And then you can talk about your clinical trial design as well. So, historically, industry has advised only using pre-submission meetings judiciously. Now that has changed actually in the last 5 to 10 years, and the FDA has become much more proactive as part of their pre-sub process. I've been very impressed with it. But previously, and you'll still hear this from people who've been around for 30 years, giving the FDA multiple chances to review means multiple chances for them to give you suggestions for things you have to address. There's also the potential for different reviewers between meetings, meaning new opinions on necessary testing from 2 different meetings. Now, the FDA has proactively agreed now to be bound by pre-submission meeting advice unless there is new information that would make the previously outlined safety testing plan insufficient. Now, the FDA does see a lot of testing on a large scale, and so sometimes a new issue occurs that they hadn't thought about in previous applications/submissions, but they become aware of it and they start looking at it. And that's typically now what I see the most often, when the FDA gives one set of advice about testing and then you come back in and they say, hey, can you do this additional test? So that is the end of topic 2. And there's really 4 questions that you should be able to answer. 1, what are the major goals for non-clinical benchtop testing? 2, what are the most common benchtop lifetime performance durability tests? What are the major goals of chronic animal safety studies? And finally, what is the major goal of chronic animal efficacy studies? So the final topic will be much quicker, but I did want to outline some of the most common mistakes that I saw and really the best practices for avoiding these. So over many years of consulting on preclinical testings, the most common mistakes that I see. 1 is not doing your testing on the full final system. Certainly your chronic animal study should be done on the full final system, as close to the manufacturing process that will be used for human studies. Otherwise, the FDA will have questions about whether it's going to be predictive. Another mistake that I often see is assuming the FDA won't require chronic animal testing for using an existing neural interface being used in a different context. Because they're worried about wear and tear, local safety issues, and side effects that may occur, taking an existing DBS lead and putting it in a different area of the brain for a different purpose, or putting it on the spinal cord, or putting it by a peripheral nerve, those are all things that have different safety risks, and the FDA often will still require a 3 to 6 month large animal study, and that's something that should be talked about in a pre-sub. Another one that is very common and very unfortunate is many people do their non-clinical / preclinical testing before they do their failure modes analysis. You should do the failure modes analysis from the beginning. You should be as hard on yourself as possible, and then each test before you do it should really be addressing those points. That's the way it's intended to be done, and you put yourself at risk if you don't do that. I mentioned this before, but a huge mistake that I saw commonly was not planning to run a short non-GLP chronic animal study with the CRO prior to GLP. And then finally, the most common mistake I see, especially from academics, is deviating from tried and true methods. You really need to do this only when absolutely needed to accomplish your goals. 6 to 8 animals at 6 months does not recapitulate all the safety issues you will see at scale. There's a reason why 900,000 pacemaker human implants a year help address a lot of the knowns unknowns. So for the FDA to be as comfortable as possible, you really want to change as little as possible and only change the things that are critically needed to create an improved effect. So in terms of best practices, again, plagiarism is a great thing in translational neural interfaces. Get to know the FDA website. So in addition to the guidance I pointed to you and some of the papers I showed you, the FDA has the existing 510Ks and PMAs for all the devices that they've approved. They are available online, and those outline, sometimes in great detail, the safety testing that was done on the bench in animals, and then the testing that was done in humans to support the 510K or the PMA. So you really one of the first things you want to do is find devices most similar to your own and use these as templates for your failure modes analysis and to seed your testing plan to mitigate. It's also an important thing to actually look up these devices for the reimbursement codes and see what prices and what range you can get, because that actually affects how you develop the device to create a business case as well, and is often overlooked. What's really interesting is, so the FDA, as part of these decision-making processes, will often get outside experts to weigh in on the data as well, and these are often public. So you can see the FDA talk about it, and outside experts talk about the decision-making process based off of all the data on the FDA website. There's often slides and videos, and these are, again, public forums where you can often go to them, and I have gone to several for neural interfaces, and they're incredibly enlightening. Some are available online now in COVID as well. So for that final topic, really what I wanted you to learn was what are some common mistakes made in non-clinical / preclinical testing so that you avoid them. What are some of the documents available on the FDA website that could be used to help your own safety testing plan. So this is an overall review over all 3 topics, and really the key things you should take away from this. 1, non-clinical benchtop and animal testing is intended to characterize potential failure modes for your device. Everything stems from the failure modes analysis. 2, FDA guidances and the FDA website and select papers are all great templates for seeding your failure modes analysis and subsequent testing plan. 3rd, benchtop and animal testing does not remove all risk from human studies. It's an impossibility for some devices. It just helps better define the risks to determine if the risks or the reward outweighs the risks. And then finally, although definitive safety testing needs to be done under GLP, and it needs to be done with as close to as possible the final system that will be tested in humans, manufactured using the final manufacturing practices. So that's important, but not all testing needs to be done in large animals, and you don't always require large animals or a 6-month large animal study. All of this follows a common sense principle. You can have a conversation with the FDA. You can state your case as to why this is the most appropriate data to predict human safety and efficacy, and they will respond to it. It's often easier to go with tried and true methods, but that doesn't mean you don't strategically propose something else if it doesn't make sense for your own device. So here are some other learning opportunities. I previously talked about the FDA IDE guidance. This is quite long, but it's quite good. Also, as part of a collaboration with MDIC, the FDA, and the NIH when I was there, we actually published a blueprint for early feasibility success through medical device innovation consortium, and so the link is available here. And finally, I'd like to acknowledge all the people who have really helped my process in terms of learning about translation of devices, both through Europe and through the FDA. Several members of CVRX were really important as I made my first transition from academia to industry. Adam Cates, Eric Lovett, Paul Pignato, Brian Soltis, and Dave Alm were all the ones who taught me, and most importantly, the wisdom of 20 years of experience in translation is impossible to recapitulate. You need people like that on your team to be able to identify all possible failure modes and their solutions. The FDA, I've been able to work very closely with, and there's several individuals who have really helped me understand the FDA process, including Kristen Welly, Pasha Takmakov, Victor Krothammer, Carlos Pena, John Doucette, and Tim Marginin. And finally, several people at the NIH were instrumental in creating these MOUs with the FDA so we could have these conversations about what the FDA is thinking and then get that out to the field. This includes Stephanie Fertig and Nick Langhals. Finally, thank you. If you have any questions, I'm happy to answer them. You can follow me on LinkedIn and Twitter. I'm also kip.ludwig@wisc.edu if you have any questions. Thank you very much.